Today here, we are very happy to host another SCIENTÍFika sessions. Some of you might know that these are our weekly scientific seminars where we interact with previous and future Max4 users. And I am Dariana Orbanic, I work in the user office, and today we have the exceptional privilege to collaborate with ESS in hosting a seminar by Professor Ada Yonath, a Nobel Prize laureate in chemistry in 2009. And her talk will be chaired by Dr. Christine Darbe from the ESS. For those of you who might not know and may be joining for the first time, fika is the Swedish definition of a coffee break. And we wanted to use it in our name to reflect the spirit that we would like to have in this chat. This is not supposed to be a formal seminar where we just listen to the speaker, but we would like to have an open discussion and ask you to participate with your questions. As usually, fika is an easygoing chat over about any type of topic with a cookie and a drink. There is also a hidden word in the name, which is científica, the Spanish definition of a female researcher. And we want to take advantage of that to mention that Max4 strongly foments the interest of female students in STEM-related matters. If you have any comments, please reach us through the email of the user office at Max4. On the technical point of view, we would like to ask you please to rename yourself, adding a name, surname, affiliation, and profile to know who are we talking to. And please keep your microphone muted during the talk so the speaker has no noises, and if possible, keep your camera on so she can kind of see the audience. During the chat, during the seminar, please, you can use the chat box to put your questions there and react with the reaction buttons offered by Zoom. And after the talk, we would like you to use the raise hand option in under participants on the bottom, so that the chair can invite you to speak up and tell your question directly with your camera and microphone on. If you cannot talk, then you can again use the chat and we will address those questions too. Finally, as Christina mentioned, we have a forum where you can also interact after this talk, and we can have this discussion ongoing, and you can contribute with your thoughts and comment also on others people's questions. Without further ado, get a drink and a cookie, the usual equipment for a fika, and enjoy this talk. Today we will discover how do intruders take over their hosts, and I leave the floor to Christine to present our speaker.
Thanks a lot, Dariana. So indeed, today we'll give a bit more human touch as well to the SCIENTÍFika platform by focusing and discussing with the most relevant Nobel Prize winner for light source and for neutron source user. So Professor Ada Yonath, who is an Israeli crystallographer, best known for her pioneer works on the structure of the ribosome. So many thanks to have connected and for this event, for this scientific event organized in collaboration between the Max4 and the ESS, and with the contribution from several other worldwide organizations, so like the Forum of International Physics and several others that we can mention later. So an educated workforce is the way forward and paving the highway to science. So that was excellent image from this morning ESS staff meeting. And we are working hard to construct this most powerful neutron source in order to complement this brightest light source that we have here, so at the Max4. So all of that for science. And the quest is the understanding. Isn't it what drives us also as a kid? So following our curiosity and our inspiration, it can lead to discovery, applied to machine, to human, to the nature. So nowadays, to observe nature, we have giant telescope and we have large microscope, like those light source, those neutron source, or more versatile, the cryogenic electron microscope. So even if we are still at the dawn of understanding the whole complexity of life, days after days, innovation after innovation, curious and perseverant scientists and engineers open new gates in the field of physics, chemistry, and biology. So as students, we often have a role model to guide us, or at least we work hard steering towards our ideal star. So today we have the honor to have among us a role model and a star, so Ada Yonath. So Ada had kindly agreed to give a brief presentation on her work and especially to chat with us. So her magic appearance on the SCIENTÍFika platform so permit to gather an impressive number of renowned scientists and new star to be. So from different institutes and different countries, from what we see. So Ada Yonath was born in Jerusalem. So she didn't have an easy childhood, but thanks to her curiosity and dedication, she became the first Israeli woman to win the Nobel Prize and the first woman for 45 years to win the Nobel Prize in chemistry. So she is the current director of the Ellen and Edmund Kimmelman Center for Biomolecular Structure and Assembly at the Weizmann Institute of Science. So while working on ribosome structure for over 2 decades, much before her Nobel Prize, so she was using all suitable synchrotron. She was among the first to use DESI for its high resolution. The Max Planck created a research unit for her in Hamburg. And from Hamburg, she was driving to Sweden, mainly to Göteborg and Uppsala and Stockholm, and later to Lund, so where Professor Anders Lija moved to visit Max, and then Professor Salam Al-Karadahi joined. And I would like to emphasize also her commitment to promote science as a vector for peace. For instance, with the Sesame initiative, so that she kickstarted in 1997 in Milano, where Gustav Wolf from DESI and Ermak Winnick gathered key scientists around a table. But the route is long, and with or without DESI, with DESI, UNESCO, and CERN, so that was a long way. And still, Ada and Erkan app that you see here in the audience has promoted this light sword in the Middle East, thanks to government and the different, the direction of those governments. And then as Erkan, I will quote you as well there while you were speaking about the Sesame to say that it was a long way, but it never died because it was a good idea. So we can see, or you can know more as well of those origin of the Sesame that brings light to the Middle East by following the Physics Matter colloquium series, which is supported by the Forum of International Physics at the RTS. So like African light sword, the Sesame keep the same type of leap motif, like the science for development. And I know that Simon, so Simon Connell, who is here also in the audience, the chairperson of the African Light Source, could give also more engagement as well to this discussion later. But now, so let's listen to Ada and think on how science can transcend boundaries. So if you can, I don't know if you want to share screen, Ada, or do you want to directly engage in the conversation?
Screen, and I want first of all to thank you for inviting me, and second to admit that we met, of course, we met by Zoom in a physics conference on a session that was dedicated to synchrotrons, although I didn't know it in the beginning until I heard actually about Sesame, but it doesn't matter. So I understand that I'm invited now to Lund mainly by the interest of physicists that they want to are busy on working on synchrotrons and the neutron diffraction. So, but my lecture in the beginning, please forgive me, the biologists, there are some very simple slides. So I will start now to share. Do you see? Do you see the share?
Yeah.
Yes.
Just maybe in full screen.
Yeah, I will try. I'm trying.
Yes.
Now
Perfect.
Almost.
Yeah. Mine.
Now it's even more perfect, isn't it?
We can. That's the perfect mode, the best possible mode.
No, something is wrong. Wait. Just a second.
We see your cursor as well, so it looks to work. Huh?
Okay. So I have a question. How do intruders take over their hosts? And I want to tell you right from the beginning, I don't know. This is a question that actually I'm posing to you. Maybe you know better or you understand better. But let's start from the beginning. Actually, before I start with the science, I want to say that most of or large part of the data that were used for this lecture were collected in synchrotrons, including the SAREF, DAISY, APS, SRL, and even KK in Japan. And recently, data were obtained by high-resolution cryo-EM. And the difference between the two techniques, I won't talk about it today, but I only relate to one point, which is time. The time that they took with synchrotrons, although we worked very hard and we were two groups, took 20 years minus 2 weeks to get the first structure, and then a few years for next structures. Now we get a structure by a few months, sometimes even a few weeks. And this is because the technique is different, but also because there is a very good starting model from the crystallography. So I used a kid's book for the beginning, describing, I think it's a wonderful kids book, children's book, describing all types of proteins that are needed for our life, like insulin and antibodies and collagen and hemoglobin and so on. I want to say that in nature, structure means function. Here there are two paper clips. They can make their function, stick together several pages. And in my own fingers, without boiling, without cutting, without acid or anything, I change their structure, just their fold, and they are not functional. And this is correct for proteins. The fold of each protein, which is carefully designed for fulfilling its function, is determined by the sequence of its components that are called amino acids. So you can see here a protein with a substrate that fits exactly into its place. If it was too small, too large, it won't fit. And if it was too small, it will jump. So the protein, this protein that cuts over here, could not cut. Sorry. Something happened. So the sequence of the building blocks, it means of the amino acids, which enable the functionality, is dictated by the sequence of the gene that codes for the protein. The DNA is collection of genes. There are other things too, but they mainly genes. It is a 4-letter language, and each protein has a code there. And the 4-letter language is coding for the amino acids. There are 20 of them. Each triplet has an amino acid. The proteins are long chains, and the fold is carefully designed. I talked about it earlier. Sorry to repeat. Okay, so the process of translation, DNA, which I mentioned a minute earlier, is a double-strand molecules, and the letters are called bases, are between the two strands, and they are not available for any reaction or any reading or anything. So the first thing that happens is the information in the DNA is being transcribed to messenger RNA, which is also a nucleic acid, but it can exist as a single chain, therefore the information is available. And this can be translated to proteins by ribosomes that are functioning the same way in all living cells. Their size and their some interactions have been changed with evolution or with specific needs, but the decoding and peptide bond formation are fully conserved. So there is a huge number of ribosomes that function in each living cell. Mammalian cells can contain millions, like in the liver, 5 millions. And even bacterial cells contain up to 100,000 ribosomes when they are multiplying. Their ribosomes perform their two major functions, as I said earlier, decoding peptide bond formation the same way. And they act very fast. They can make 40 peptide bonds in 1 second and hardly make mistakes. In a more complicated systems like human, they make less, 5 or 6 peptide bonds in a second, but they hardly make mistakes. I, as a student, had to make a peptide bond chemically. Took me 6 hours, and I was the fastest in the class. 6 hours. Mistake rate is very low. They hardly make mistakes. So we like to describe them as factories that are made of two floors. The top floor is where the information is being decoded here. And the lower floor is where peptide bonds are being made. So when a triplet that is coding for this amino acid is being noticed here, each amino acid has its own triplet, then this amino acid will come in and be connected to the newly born protein. This happens inside the factory, but I don't know how to draw it. So I drew it this way. The protein will be longer by 1 amino acid. The messenger will progress in 1 triplet. And this particular track, which is also an RNA molecule, a tRNA, will come out with the tracks and go on. That's all. It needs only very little energy. That's all what happens in order to make a peptide bond or peptide bonds. Just to show you the track, the tRNA is a molecule that is always L-shaped, and all through evolution and everywhere. There are some changes in the sequence and in the fold, but it's more or less the same with an anti-codon loop here. It can make base pairing, like in DNA, between this and the messenger RNA, and the cognate amino acid that is bound to it in the lower part, which is fully conserved CCA. And this happens in the position where peptide bond is being made, which is called PTC, peptidyl transferase center. So it took us 20 years minus 2 weeks, to determine the structure of ribosomes from bacteria. But when we did, we immediately understood how they work. So here it is. Small subunit is standing and waiting, and messenger comes to it and goes in puff. How does it come? This is an open question. But when it's in puff, its position and its direction are monitored by an initiation factor, this blue thing here. In bacteria, there are 3 of them, and this is number 3, that that makes sure that the messenger is in place. And when everything is fine, the first tRNA can come to its position. There are 3 positions. I won't talk about it much, but it is brought by another initiation factor 2. Here it comes. The factor is in light blue. The tRNA that brings the amino acid is in orange. The amino acid is this little thing here, this little blue thing. And where everything is fine, the large subunit can come, the factors go away, and the 2 subunits associate with each other. And now we have an active ribosome, and we look at it from the solution. On top is the large, below is the small. And amino acids are being brought by tRNAs with elongation factors, these blue things. And they come in, the ribosome helps. We took away the large subunit now. You can see how peptide bond is being made, how decoding happens here. And peptide bond in the lower part. And from the whole large subunit, we left just the tunnel for which the protein goes out. Now you can see the whole ribosome again. The tunnel was here and the protein, this blue thing, comes out and folds here alone or with chaperones. And the whole process continues until there is a stop codon. It means a codon that says protein is finished. And then instead of elongation factors come a factor which is a stop factor or a recycling one, and it comes in, release the two subunits. The protein can go and do what it needs, and the whole system is waiting for next job. That's all. Isn't it clever? That's it. Very clever. So what we saw is that ribosomes are built of 2 subunits that associate to a single particle for the translation, and the small subunit decodes, the large subunit provides the position for peptide bond formation. You saw in the movie something very fuzzy, but actually we know the position of every atom in the ribosome. Ribosomes are made of ribosomal RNAs, it's the gray, and many ribosomal proteins, which are in all colors. This is the small subunit and the large. Now, more of them are known. Not only bacteria, also of human, of other very complicated cells like mitochondria and so on. Just to remind you, the small makes the decoding and the large provides the position for peptide bond formation. And this looks like this. If we want to look at it like the factory, on top, the small subunit with positions for decoding of 2 tRNAs and 1 that is exiting, and the position where peptide bond is being made. That's all. Very clever. And again, to remind you, top, small subunit, large subunit, and tRNA in between connected. So now we understand, I hope, how proteins are being made, or at least the major points of it, the major sub functions. Let's go to the intruders, the invaders. And the question is, how do they take over the replication system of the cells, the invaded cells? So the invaders are viruses. You can see EM electron microscopy of the one that everybody talks about, COVID 2019. And you can see here lots and lots of viruses. Ours is one of the smallest, yet it created problems in the whole world. So what's really happening? First, about the history. So the first SARS was discovered. SARS is the name of the family, was discovered in the 60s, and it was for avian infection. Then it infected chicken. Then came the coronavirus. The first coronavirus came already was actually over already in 2002 with what was called at that time a large number of cases, 8,770 deaths. Now it is a very minor number. And just to say the name, it's severe acute respiratory syndrome. These are the sources. In 2012, it was found in Saudi Arabia, and then there was a MERS type or MERS subgroup that was a Middle East respiratory syndrome that was a bit more severe than before, but still, in our terms, not severe. 34% deaths of the cases, but all the cases were less than 3,000, 2,500. In December 2019, in Hubei province in China, in Wuhan city, there was a new coronavirus, which is called now COVID 2019, which is the one that causes the coronavirus disease. So as you saw earlier, let me show you again. This is a cut, artistic way to draw the cut through the coronavirus, perpendicular to its round shape, which shows that it has here what we call spikes, which are those that bind to the surface of the cells of the hosts, like human beings, and can sneak inside, can bind strongly and sneak inside. I won't go part by part. I will just focus on the genetic material. The genetic material is RNA. It doesn't have messenger RNA, and it is what is called positive RNA. It means it just can go in and be translated. Does it really? So, like all other viruses, they are parasites. And they can do many things for themselves, but their replication requires host cells. And although the size and the composition and the complexity and the functions encoded by the genome are remarkably diverse, all viruses rely entirely on the protein-forming machinery of the host cells. Thus, in the absence of their translation machinery, they must recruit cellular ribosomes, host ribosomes, in order to translate the viral messenger RNA, in order to produce the proteins required for their existence. And here comes one of my questions. Do viruses use host cells procedures to make their own proteins? It means, can they intervene with the host cell procedures? Or do they just jump into the already functioning line with the virus messenger RNA? In my opinion, both answers are not simple. So let's look for a minute at the genome of COVID-19. It's a single long RNA chain, which is coding for a main protein and secondary proteases that cleave the main protein in order to produce the needed molecules. So here it is, this long, can you see this long line that I don't know why, but in all books, in all papers, they draw it this way. Not so important how they draw it, but important what it is made of. So this whole long line is one line when it is in the virus, but actually it contains proteases and frame shifting. I'll come back to it, and many other things, but I will focus first on the viral spike, the spike that that goes into the cell. I'm focusing on it because we know how it's built. This is this one here, and it's built like this. There is already a structure. Actually, there are structures of the spike of several viruses, but the one we talk about now is COVID-19. From the side, it looks like this, and from top, you can see here a place that it can jump into the membrane of the host cell. This is it. If you want to go piece by piece, this is it. The part that is supposed to be or found to be its gene. Let's look about a little bit more about the virology and now focus on the frame shifting piece. We already talked about this part, which are the proteases, about the spike. Now the frame shifting. This is very important for getting into the ribosome, for the virus to continue. So the structure of and the mechanism of the ribosomal frame shifting during translation shows that in all viruses, program translation frame shifting is conserved. Doesn't matter which corona, they have the same type of frame shifting. And this is necessary for the production of several viral proteins, including the viral mRNA proofreading, because they also need to make sure their proteins are correct. So here it is, it's here. Now, I want to focus on another protein, NSP1, which is a ribosome gatekeeper. A protein that can keep the gates of the ribosome. So if you remember, ribosomes looks like this, although this is bacterial, but in principle, small subunit and large subunit. Until now, I drew them up and down because of the factory, but actually they can stand on the side. The PTC, where protein is being made in the large subunit, and decoding happens here. It means here is where messenger is, and tRNA connects the two of them. So it was found that NSP1 can interact with this part when the small subunit is alone, before the ribosome is being made. So you can see here a figure from a paper by Tidu about it, the gatekeeper, and you see this is the small subunit, a human small subunit, and messenger RNA will come here. So the gatekeeper goes here, and it has the ability to bind the messenger of the virus and create a specific structure of it. I won't go into detail, but this structure eliminates the possibility to bind other messengers. Wonderful. So what? So the messenger is bound. So what? What now? Actually, if we look at it, oops. Oh, sorry. If we look at it a bit larger, it is sitting here, the small subunit, and messenger should bind here. That's also where the disturbance is. The gatekeeper binds, or actually this NSP1 binds here, and then binds. And even if messenger of the virus bind next to it, so what? That's not enough. In order to make a ribosome, factors are needed. Initiation factors. I showed in bacteria 2 of them, 2 out of 3. But in human, there are more than 20. How will this thing attract the factors? This I don't understand. Only when it attracts the factors that it can afterwards bind to the large subunit. But how does it happen? How? It's not enough to bind here and block. The cell does not want blockage, it wants working. And it recruits initiation factors. They are not just floating around. It recruits it when it needs to produce a protein. How does it happen? Big question mark. Actually, this is the question mark. How do these intruders take over their hosts? This is my question, and it's still a question. And I'll be really happy. There are some ribosome experts here. I'd be really happy if anybody can tell me, because it not understanding it disturbs me. So essentially, this is the time that I can start my credits. I want to thank the Weizmann Institute that let me work a long time with hardly any mistake. When I was called a dreamer and a fantasionaire, the person that made the decision was the president, Michael Saylor, who is a biologist, and the Weizmann say that they let me work in a very low position and without a future expectations to develop. But anyway, I could work. It was actually the best time that I was, just nobody cared. So they said that he did it because he still is a biologist. But when his term was over and he was replaced by the next one, Chaim Harari, who is a physicist, he let me go on. And they were both advised by the advisory committee that included Sir John Kendall, that determined the structure of the first protein, Nobel Prize winner. Chris Anfinsen, that shows that ribosomes have to have structure, also Nobel Prize winner. And Alex Rich tried to determine the structure of ribosomes for 10 years and failed. The three of them were supportive, although they said it's a very important project, shouldn't be stopped. But the chances that Ada make it, no, not very high. It all started with a strong collaboration with Professor Wittmann from the Max Planck in Berlin, who was so interested in seeing the ribosome that he even helped to start the group that was described earlier at the DAISY. But he died 10 years before the structures were determined, and he was replaced by Franceschi and then by Fucini. So I had two research groups, and I want to thank them for their devotion and determination in good and in bad times for many years. I want to show the group in Israel. It's actually, most of the people here are already gone, already left. But still, I want to highlight Anat Bashan, who is the head of the group, who is Dr. Anat Bashan, the senior person that determines what everybody else does. You can see here who financed and which synchrotrons we used. And Tamar. Tamar came 19 years ago for 10 weeks. She's still around. And the day that I took the picture, she had birthday and she baked a cake. You see the cake? Probably you don't see it well enough, so here it is. So in my group, the cake, the ribosome is considered sweet. I also want to thank my family. I can't show my mother, my sister, and so on, but I can show my daughter, who is an MD, a director of a department in Sheba Hospital, the best in Israel, and her daughter, who is now a student, medical student. But at the time that I talked about is when I got a prize in 2008 in Paris, and she gave a speech. And she said, as you know, she's a very busy scientist, but she always finds time for me. And I invited her at the age of 5 to the kindergarten to explain what are ribosomes. And she gave me a prize. This is not her prize, this is the Nobel medal. For me, her prize is as important. The grandma of the year is Ada Yonath. So I asked her which year. Every prize has a year. And she said, which year? You have to prove yourself every year. And if you fail, I take it off the wall, but it's still on the wall already since 2008. So 13 years. I'm still a good grandmother. It became popular in Israel. This is a carnival. He's me. You see, it's him and his wife. When they're dancing, you see that she's producing a protein. And even small kids become ribosomes. We also want to thank Michel Kishka, one of my most appreciated artist and writer. He made this picture of me. He's not a scientist and he didn't know me, but look at it. Large subunit, small, small, large, large, small, small. And he thinks that this and this and this and this are positions for new antibiotics. Thank you.
Thank you so much. So the head field of ribosome. So that's a very nice image. Huh? So thank you so much, so Ada. So now we really understand how you can stimulate young scientists as well. And this true science interest for everybody and to turn on the imagination of the youngster. So thanks a lot. So we have, so in the audience, many students actually, and we see that there are quite some questions already that pops out. So we have as well our special guests here. So with Professor Anders Lija and Salam Akaradida, sorry for the pronunciation, and also Michael Eriksson, who returned virtually to Max 4. So it's really nice. And it's really nice as well to have my colleague from the Forum of International Physics at the American Physical Society, so Alan Burt and Luisa Cifarelli, and also Bill Barletta, who are here. So, and together as well, as we mentioned, so with Erkan Alp, and we have as well from DSRF, so Ed Mitchell, who is there. So we can certainly as well have a grand tour as well of the light source and have possibility to ask more questions. So maybe if it's interesting or possible, we could start by question from students, because I think it's a nice way to first engage them. So you have a chat box there. So if you want to ask your question in the chat box, and Erko will help me as well to address them. So you can enter, or you can put your hands up, and then we will ask you directly the question live so that I can answer. And we can start anyway as well with some question that we have on this wall of ideas. Or I would maybe ask Garima, because I know that you connected from India, and maybe you can as well share your microphone and ask live your question. Okay. Yeah, maybe if you can stop sharing the screen, Ada, like this, we can...
Trying. How can I do this? I'm trying.
On the top right, maybe, there should be some button pressed. Voilà, exactly. Excellent. Now, so we see everybody. So I don't know if, Garima, I know that you had this question, or should I read it? Because I know that you put it as well on the wall of ideas. So maybe the connection is not easy to India. So then I will just read as well, because she was reading so your biography also, and then was mentioning on your personal path, actually, if it's okay that we speak about your life as well. So because you said that, so your mother struggled in your journey upon here, and it was really inspiring, so for all of us. how did you manage to keep up the spirit alive and in all those situations? Also, when do you say that Marie Curie is not your role model? After reading your biography, can you elaborate on that? I mean, I'm French also, so then I was also curious to know why.
First of all, maybe I was good in ribosomes, but I cannot get out of my, I cannot see you.
Yeah, we can hear you. It's all fine, yeah.
It's enough that you see me. I also want to see you.
But the ribosome is better. I mean, your head full of ribosome is better. So you can just think that we are behind your head.
You see this?
No. We are seeing, we can see everybody, so it's okay. But then we can.
Okay. So you ask, how could I go on?
Just
I'm curious. My reason was curiosity. I was really curious to see how things are going, and I didn't care for anything else. That's it.
so mainly. So do we have maybe anybody would like to?
Christine?
Yes.
Christine,
Can I ask
there
a
is
question, please?
from Kyoda Dada.
Exactly.
Yes.
Can you repeat? Yeah, I didn't see because there is too much bright light. So yes, please.
Okay, sorry. My question
Shall
is,
I answer again?
can you hear me?
Yes.
Yeah. You actually talked about the intruders. My question is, hello? My question is, can the movement of the intruders be determined? That is, the way they move at the host sites. Can you actually determine their movements?
I didn't understand the question.
I mean, can you actually determine the movements of the intruders as they move through the body to the host site?
Yes. Okay. So what is the question?
Can you hear me?
Yeah. Okay. So the movement of the intruder to the host site.
Yes. Can that be actually determined better?
I don't understand it. I don't know everything. But there are many questions. But my more important for me is how the ribosome becomes how they convince the ribosome to work for them.
Okay.
How to convince them? So this is an open question. So this is really why we need as well new scientists to sort this out.
Yeah, this is a question that I'm asking myself, but also you.
So then are we getting ready for that? How do you go there? So with light source, with neutron source, we have some thinking to do then.
Sure, if you want to.
Yeah, so this is a good exercise. I think we will.
If you can answer it by the light source, will be fantastic.
And neutron.
Okay, I was actually thinking if we can stop the intruders before they reach the site, the old site, it will be very nice.
Indeed. So we have already few light source that could start with the crystallography to answer those questions. Neutron will come. And
You
then
think that you can answer it crystallographically?
I don't know. What do you think? Because now with the resolution, this is still the best we can do, isn't it? Or are you thinking about with the cryo-EM then? This is what you would rather go to?
I don't know. If I knew how to do it, I would do it.
Good.
We have a question from Anders.
This is why I'm asking you.
So
if I had an answer or if I had an idea, I would do it or try to do it.
Anders has the answer, I'm sure.
No, I don't. But I'm very keen to understand how far you got with regard to this interesting gatekeeper protein, NSP1, because that seems to be a very crucial element here. And
correct, it's not my work, it's from the literature.
Okay, but anyway, part of the secret is residing with this protein. how can it discriminate between host messenger RNA and viral mRNA? I think this is a crucial question.
Yeah, you're right. I think that the ribosome cannot distinguish, but the ribosome is not, cannot be formed without the cellular components that make it, that makes it being formed from its two subunits and so on and so on. So how does the virus make it? Once it makes it, it has the gatekeepers. That's fine. This I understand. But how is it being made? Or does it jump into already made ribosomes? How does it do this?
Maybe there's enough copies of this NSP1 protein with the virus such that it can get to the ribosomes of the cell and prevent further activity of host messenger RNA.
You're right, very right. This was my first thought. Maybe there is enough of it and it's very active, but what happens then? It sits there and it's very active.
Well, it's very interesting.
It prevents the cell. This explains why the cells that the virus are inside, they are suffering. This I understand too. But how the virus convinces this machinery? And there are millions of ribosomes in the cell. I said it in the beginning. Maybe it conquers only half of them, but still it has to conquer them. And there's 60 years in ribosomes and it's not enough.
There's another interesting question coming up here.
Yeah.
And the virus goes in within seconds and that's the thing.
There's a time factor as well. So simulation maybe could find ways as well to explain in different disciplines.
Could be if you make it anything that you think can answer this.
So
but maybe you have other questions, not only my question.
Yeah. We have a question from Ipsita Banerjee.
Yes. I don't really have a question, but I just wanted to say that it's so nice to see Ada again, because I actually saw her like 4 years ago when I was a visiting student at Professor Debbie Fass's lab at Weizmann in the Kimmelman building, and I have very, very nice memories of going and carrying out measurements from like in her lab, which was right next door. So it was really fantastic to see and hear her after 4 years.
Thank you.
Thanks a lot, Ms. So we have maybe question about the simple global warming. So there is one question, we don't know exactly who put it, but then so much about the macromolecular crystallography having transitioned from room temperature experiment to cryogenic ones. So what do you think people need to rediscover and learn about the sample when methods such as serial crystallography make room temperature data collection more accessible? What do you think about that?
Crystallography at room temperature? Why not, if the material is not deteriorating? I think you can do it on sodium chloride or in other salts, or even more complicated salts. But on a biological sample, the sample is not alone there. There is water, there are solvent and salt, and at room temperature, all these things are running around and may disturb the crystallinity. But you can, if you find the materials that don't suffer. Many structures were determined at room temperature, but not many biological structures.
This is a challenge for Zoe, maybe, huh? And ESCO.
Excuse me?
So we have ESS scientists as well here who are developing some science environment as well, or maybe in Los Alamos as well, they could help. So to try to take this challenge as well.
So maybe I can comment on that. So we actually regularly now collect data at room temperature, just taking a big enough crystal, so then we can distribute the dose on a larger volume. So that nowadays works well actually.
Crystallographically?
Yes.
Good. If you can do it, why not? It's much more friendly than at cryo temperature, and there is less worries.
We sometimes see a difference due to the cryo temperature, so there are minor changes.
Why not? Of course.
Very good. So maybe, oh, I see that, so Salam Akaradai, so you have a question as well.
Yeah, again about this NSP1 protein. I wonder if, is the structure known for that protein, Ada? For NSP1?
Yes, I think so.
I
can check it. I think so.
I just thought that is it somehow would be interesting to check if it's somehow related to any ribosomal protein structure, that it somehow mimics ribosomal proteins, and that way it binds to the ribosome.
It's a good idea. I will check. But the factors that bind to the ribosome in normal cells, especially in eukaryotes, in human, there are so many factors. There are over 50 factors.
Yeah.
Just for every amino acid, the elongation factor and all the rest. So most of them are not like ribosomal proteins. Ribosomal proteins have hardly any structure without the RNA around them.
Yeah, I just following Anders' question about this recognition. How does it recognize or distinguish between these two RNAs? Could be some fingerprints for recognition could be recognized. I don't know.
You're right. Something has to be recognized, like you call the fingerprints. But for me, this gatekeeper is very important, but it doesn't answer the whole question.
No.
The entire question.
No, of course not. And it's like the story of the egg and the chicken who comes first. And because the virus has to synthesize the NSP, the gatekeeper, somehow before the whole process starts. And if it's not present in the virus, and then it has to take over somehow to push the gatekeeper into the synthesis machinery, and then the gatekeeper would go and block the ribosome. How that happens, that's what's interesting.
Correct. You phrased it in another way.
So it's multi-function, multi-physics, multi-biology, multi-phenomena. So that certainly has to take into account. Huh? So we have one more, several other questions, if you don't mind as well. So for instance, if you could speak a little bit more about the environmental issue linked to the antibiotic resistance and how it's challenging to create antibiotic is, so especially as well when they are biodegradable. So biodegradable antibiotic, and then why is it challenging?
So this is a topic for a long discussion, but in a few words, the antibiotics are, most of them are weapons of bacteria against another bacteria, another type of bacteria. And we are using those that are not poisonous to human beings or animals, and there are many of them. Half of the antibiotics used today are targeting ribosomes. So if you want to get rid of a pathogen, you stop its ribosome or you disturb the ribosome, and the pathogen within a few days, few generations will disappear. This is the way the antibiotics work. But the bacteria are clever and they want to live. So they found ways to have resistance. There are many ways for acquiring resistance. Some of them are just taking advantage of accidental mutations, like the mutations of the virus. Yes, there are mutations all the time, and those mutations that help the virus be alive when there are antibiotics, those exist, those remain. Others are more complicated. There are also some pathways to make antibacterial materials that catch them or influx. I cannot talk about all, but those that we understand on ribosomes were improved by companies. The first 20, 30 years of antibiotics was fantastic. They were improving every known antibiotic that was not poisonous. They made it faster, quicker, more efficient, whatever. They didn't produce many new ones because it seems that nature produced most of them. I think there are, if I'm not wrong, 2 new, fully new type antibiotics, ribosomal antibiotics that were found to be clinically important. So what we are trying to do is to target the ribosome from its periphery, from the outside. All the active sites are in the center. We are trying to target the outside in a fashion that is pathogen specific. It means we look for motifs on the outside, on the periphery, that are specific to pathogens, especially to resistant pathogens, and we are trying to target them. And we already showed that we can stop the life of bacteria by this targeting, probably because we are disturbing interactions between the ribosome and the cell, not within the ribosome. But this is not straightforward. We have those that are targeting and stopping their life, but we are not sure how they bind and how to improve them. It's a lot of work. And we are connecting to them moieties that can penetrate through the cell wall of bacteria. So we do this, but when they penetrate this way or that way, just to tell you all the difficulties will take me more than an hour. So let's stop it here. You understand the magnitude of the problem.
Yeah, it's a big problem. We can see that definitively. So we have another question about synchrotron. So how can synchrotron help with finding solution for the current pandemic?
If the synchrotron can determine the structure of beginning of conquering the ribosome, virus goes into a cell and begins to conquer. Maybe then we understand. But I'm afraid that synchrotrons are not the right machinery for it. Not that I have the right machinery, but
Can you expand on that, Ada? Why the synchrotrons are inadequate?
For this people, they are adapted for many things. Don't worry.
No, I know, but I mean for this particular purpose. Why did you come to that conclusion? Because
as far as I know, synchrotrons are producing crystallographic data or low resolution non-crystallographic data. No, low resolution is too low, and crystals, I don't think you can get a crystal of a ribosome with the intruders or the intruder species, that maybe one can. Anyway, you know, I was working 28 years on synchrotrons, and I even created or was part of creation of on the one that you are interested on Sesame, and I was on the advisory committees of APS and SRF, almost every SSRL, I was advising and judging. So I love synchrotrons and I did a lot for synchrotrons, but this I think is a problem that is beyond synchrotrons. Maybe I'm wrong.
Thanks,
I
Ada.
think I'm wrong. But somebody shows that.
But maybe as well in new techniques with synchrotron, with
Maybe, hopefully, why not?
Yeah, we can always think and hope. So maybe just two more questions that I would like to read as well. So thanks. So by thanking you as well for introducing the cryobiocrystallography to the scientific community. So journey is an inspiration to young scientists like us. So I don't know who wrote, but a young scientist. So however, with the advance, with the, well, see, more on your side. So however, with the advent of room temperature data collection, more protein crystals still required cryocooling. Could you please advise on improvisation of cryocooling when it comes to large crystal?
How to load crystals?
With large crystal, that's one of the things which is important. Now maybe with free electrons.
What is the question? How to load crystals that can be cryocooled?
Exactly. From the sample.
How to cryocool large crystals?
I think that the size is tolerable. It's possible to work on large crystals, especially if you can focus on pieces of them, so you can get more fresh data. How to load them is to find a solution that is a cryo fit solution and doesn't disturb the crystal, immerse it in it, and fish it with a loop or by a spatula, get it in between double spatula and freeze it immediately, shock freeze it. So it's not really frozen, it's glassing. And keep the low temperature throughout the data collection. What was considered impossible and not very clever, but works. Actually works very well.
Okay. We have one question as well, speaking about initiation. So once initiation begins, so that it always finishes. So the environment must be very noisy, and yet the accuracy is amazing. So 1/10 to the 6 or better. Perhaps the process start and stop. So I guess this is a question of time for the measurement.
I wish I understood the question. What is the question?
I'm not sure, actually. So what I would advise is potentially we will put as well more comment, because there's a comment box in this wall of idea, and we can ask whoever anonymous put the question to elaborate a little bit. So I think we will make the best use of this table. And then we can maybe see if anybody would like to have a question live. And there is quite a lot also to try to understand as well with the current journey as well with this virus as well, to try to find the best way as well to develop the vaccine that will be, I mean, the universal vaccine, who knows? And also somehow the advance in technology that we have nowadays with those artificial intelligence or all those machine learning, there's a lot that could definitively help as well to run and to think on finding our society some better way to connect as well with, I don't know, you always think in terms of evolution and potentially those type of cyborg. I mean, somehow maybe one day with the CRISPR, for instance, the CRISPR method, or all those type of biological, technical advance as well. Do you believe in something like this? Will one day we become bioman or biowoman, or what is the capacity that is in our genome to be transformed or to become machine maybe one day, if we extrapolate? It's not a wish, I hope, but who knows, future.
I really cannot answer the last question. I'm not a prophet, and
but it's not a world where we would like.
I can only imagine, but my imagination is not as good as yours.
Well, we have a lot of scientific, I mean, scientific inspiration and science fiction somehow. You know, this is a two common root as well, we could say. So we'll see. So what we will do really, it's really like, yeah, taking a lot of your time, but we appreciate so much as well to have the possibility to listen to your still open question. And I think this is like some challenge as well for us and for the youngest generation that listened, and hopefully that will find some solution in 1 year, 10 years, hopefully not 100 years, because we'll not be here anymore. And we'll see as well and stay tuned to all of that. So do we have more comment maybe, so from your colleague or from understanding from the, I mean, your initiative as well, it's really recommendable as well. Every aspect that you have done for the step for the Sesame, for instance, or all your way to use also physics, I mean, and science to unified, so through religion, through all this aspect of political diversity as well in the Middle East. So you really inspired and help a lot to think and to have science like one of the driver for connecting and unification of all those causes that are very noble. So definitively, it's just like this peptide somehow that you were describing in the protein, or like the hydrogen bond. You know, it's just like brings science being like the link to all our society somehow. It's maybe like a strange analogy, but so somehow I like to think that science is that important. And thanks to this kind of ribosome, this possibility, this way of thinking, this machine that you best describe, and that is still to be understood from what you're also asking. So it's good to have open questions. This is why science is there, I would say. So that's really nice. So, and this is maybe, I don't know if we have more questions, or should we think on, or maybe from the SRF, or if Zoe would like, one of the two would like to say a few words, or from the Max 4. So nobody have the answer.
I have a question. What was first, the genetic code or its product?
Well, that's a good question. Do we have the answer? But we have to make some experiment to try to understand, and then we'll make.
Correct. Or you can try to use something that you can compare it to, like what was first, the egg or the chicken?
You see, this is why I like nature in general. Nature, this is like the answer of everything. It's so complicated that we'll never get the answer to that, which is good, because then we'll search and we'll go on searching, maybe forever, but we'll find little solution. And we like
I have an answer to this question.
You do?
What was first, the genetic code or its product?
So then you're a prophet.
The answer is first was the ribosome.
We should have thought about that one. That's excellent. So, and this is close to Easter, so it's perfect. We have ribosome for Easter. No eggs. Thank you so much for your sense of humor as well. So it helps so much as well, I guess, and I hope the youngster community as well to get into science and to become a SCIENTÍFika. So thanks a lot. So maybe it's good that we really collect as well this wonderful audience, and really greatly appreciate. And Simon has some comments. He waves his hands. Please, Simon.
Let me just announce that I will run some poll questions to see just a bit the profile of the audience, but please, Simon, go on with your question.
Okay. Well, I just want to say it was really a fantastic talk. I thoroughly enjoyed it. Was expecting you would answer the question that you asked, and when you were saying that function followed form, I was even more expecting it. And I was a little bit in shock when you left it open for us.
I'm sorry, I didn't want to shock you.
It's very important to have asked the question because it really highlights what you think is a very, very important question that needs an answer. And I was just thinking when you were telling the history, you know, there was one person, I forget his name, who passed away 10 years before he saw the structures that he was working towards. I thought that was very sad, but I'm sure that's how things are going to go, that there will be these technical advances and conceptual advances that will enable us to make this progress. And so I think I just wanted to comment to say, I think it's so important that you posed it as a question and left it, especially for the younger people, to focus their minds what we need to aim at. And I do see that there are always improvements in ideas, in concepts, in technology, and I have no doubt that question will be answered soon. And thank you very much again.
Thank you.
Yeah, thank you very much. So we have some, I don't know, Doriana, do you need, how does it work with those polls? So it's still okay to run, I guess, in the background? This is what happened?
Yes, yes. It was just to get a feeling of the audience and how many younger population we have or experienced researcher, but we see from the results that there is a bit of everything. So yeah, we've seen a lot of people from different profiles attended today. So yeah, thanks.
Let's not forget to take that photo that you mentioned, Christine.
Oh yeah, this is true. Did we take a picture?
I think everyone should turn on their video cameras and smile.
And we have as well with, so that would be nice. So do you have also a question? Maybe Andrea, I see that from the Sesame from the Middle East as well. So
no, more than question, I have hopes. We are still struggling for having our own MX beamline. We're strongly working on that and hope that it comes soon, so that we can join the effort.
Very good, very good. And from Los Alamos, so do you think, Alan, that there is a hope also?
Oh, there's always hope, and I think this this get together as a FICA is an exemplar for future ways to get together now that we've all learned how to use Zoom.
You see, there is always some positiveness out of all of that, even with this pandemic. I have to admit that I don't dislike that. It's just like not giving possibility to travel out of the 3D. Now it becomes a 2D world. We are even more behind screen. So we can understand as well the younger generation that way. But we can also like find ways to engage them in 2Ds as well by teaching them more. And we have a lot of online courses, actually. I could also mention that with the Max 4 and the WSS and the Lund University, we develop as well those so massive online courses to explain what accelerators are and then how we can as well so look from generation of light source and then how to apply it to medical physics as well. Medical accelerator can be used for that, and description of how we develop cavities and all those different aspects. So it's like I think very interesting to have those online tools and the e-learning, and then to get more information on how to join the scientific community. So now there is more or less no excuse. Even with the pandemic, you can always get busy for sure, and potentially one day become as well a Nobel Prize. Or win the possibility to understand how those ribosomes will really work out. We need as well to have some universal vaccine. So please also back to your your homework, and then we need simulation potentially to develop that. So there's definitively a lot to do again in this scientific world. So then we can just, or mainly as well, recall again this wall of ideas. So it's really one way to think on getting and engaging the youngest generation with the expert, and then the expert can answer to the question. So just feel free as well to use this profile, and we can definitively see how to potentially have more interaction on that. So I don't know if Michael had to leave, I think. I don't see him on the screen anymore, but then we will definitively reconnect as well all our different world. So I don't know if Ada, you want...
Thank you very much for inviting me and for the lively discussion, and for not being mad at me that I'm asking you questions instead of answering. And good luck.
But you put those questions that are so relevant. This is what is important. So this is really good. And you make us think out of the box as well. You are challenging us. So this is what is so so wonderful. Just like to keep as well those open eyes as well, and to show really that even by focusing on our daily work, so we can really look at what is happening around us and how this nature is working, and then we can find and collect as well more scientific eyes around and find common solutions. So it's really like amazing. And we are so happy that we are building as well in Lund this center of excellence in a collaborative way, in an open way, and the sustainability of all of that, so that we can find solutions for the future. So Lund is a very nice place to be in reality or in remote as well. Everything can work nowadays in remote, and I think your colleague can say the same, isn't it? That's why we stay here. So
yeah, that was a very nice opportunity to see Ada. Ada was in Lund several years ago, I think. I don't remember exactly the year. There was a lecture at the medical faculty then.
I think that I've been several times.
Yeah, but last time was at...
Maybe even before you.
Before me.
You were still in Stockholm.
Yeah, but the last time you had the lecture at the medical faculty, I think.
That's correct.
Yeah. I remember you came directly after the prize ceremony to Lund and gave a lecture here in 2009 in December. I even have a photo from that visit. So
the Swedes are very efficient. They invite for a ceremony and they give a list of 10 lectures that I have to give. All over Sweden.
You could give them online now, Ada. So that's good.
Yeah, I think that this year was online.
Yeah.
It means last December. But ladies and gentlemen, I'm sorry, I have to go.
Thank you so much for your time.
It was really nice to chat with you and to remember all days.
We will remember it forever. And this is all recorded, not only pictures, but movies as well. We'll send you the movies as well.
Thank you.
Become a SCIENTÍFika. So because physics matters.
Thank you.
Physics matters.
And thanks to our hostess, Doriana, and our mistress of ceremonies, Christine.
Thank you for participating and contributing to the discussion. We hope to have other similar occasions soon, and also to maybe meet you on site at Max4 and DSS sometime soon. I will leave you in the chat a link to our SCIENTÍFika sessions in case you want to check them out and participate in another occasion. And that's all from us for now.